Good afternoon, everyone. Thank you very much for joining us on our third Facts and Snacks Lunchtime Journal Club discussion, where we discuss COVID-19 related research and how it affects our communities. My name is Elisha Barrientos, your host and a community engagement specialist at the Research Centers and Minority Institutions, or RCMI Community Engagement Corps, led by Dr. Carla Williams, who is here with us today. So our group's primary goal is to ensure that RCMI-supported research addresses the needs and interests of the local community. To achieve this, we partner with local organizations to address health-related concerns, promote community participation in research and partnership with researchers in study design and execution, and ensure research findings are shared with relevant communities. The CEC offers consultations to Howard research teams to incorporate community engagement best practices into their projects, as well as facilitate community partnership introductions and more. I am very pleased to welcome Dr. Yousef Tizabi as our speaker this afternoon and hear more about his COVID-19 related research. Dr. Tizabi received his bachelor's in physics from University of California at Berkeley in 1970 and his PhD in physiology from Georgetown University in 1974. Following a postdoctoral fellowship at the Laboratory of Clinical Science at NIMH/NIH, he joined Howard University in 1976, where he is currently professor of pharmacology. Dr. Tizabi is an educator and avid researcher. He has over 140 peer-reviewed publications and has delivered numerous presentations at national and international meetings. He has participated in NIH study section and has been a reviewer for numerous scientific journals, including Neuropsychopharmacology, Molecular Psychiatry, and Lancet. He has trained a number of master's, PhD, and postdocs, and has enjoyed continuous funding by the NIH, pharmaceuticals, and other sources. He sits on editorial boards of several journals, including Neurotoxicity Research. He has been awarded one U.S. patent and another application pending. Dr. Tizabi's main areas of research include elucidation of neurobiological substrates of mental disorders, including depression and drug addiction, specifically alcohol and nicotine. He's also keen in understanding neurodegenerative mechanisms and exploring novel interventions, particularly in relation to Parkinson's disease. Thank you very much for joining us today, Dr. Tizabi.
It is a great pleasure and honor indeed to be here. And I want to thank you for this very kind introduction, Elisha, who has been instrumental in setting up all these details of presentation. And also would like to thank Dr. Carla Williams for her kind invitation to participate in this symposium or journal club and present some of our work. It's really a pleasure to be here. But before I start, I need to make two kind of general comments, announcements. First and foremost, I would like to dedicate this talk to Dr. Robert Taylor, who was our ex-chair and ex-dean, who suddenly passed away 3 years ago. He was really instrumental in bringing me into alcohol research, establishing Alcohol Research Center at Howard University, very supportive of research enterprise, and was a great mentor. I'm sure he has supported many people, and I really miss him. I'm sure many people in the audience have the same feeling. The second announcement is that I am by no means a virologist. So if few years, 5-6 years ago, you showed me these pictures and asked me what you see, I would have said, wow, these are 4th of July fireworks for sure. And if you showed me this one, say, wow, this is probably like those deep sea bombs that they use against the submarines that you would see in World War II movies. Well, here I'm giving
Dr.
my
Tizabi,
take. Yes.
I apologize. I don't mean to interrupt, but your screen is not sharing yet.
It's not sharing? Oh,
No, sir.
let me go back then. Share screen. How is it now?
Looks great.
But I have to make it full screen, right?
Yes, sir.
So let me go back then to show the, so this tells you how updated I am on this modern technology. Okay, so let me go back. I mentioned Dr. Taylor and his great contribution to my getting involved in alcohol research specifically, and his support and all that. And I was showing these pictures for my disclaimer for being a virologist or anything to do with viruses. Basically, I was saying if I was shown this picture, I would have thought that I'm I'm looking at 4th of July firework, and this one would be like the deep sea bombs that you would have against the submarines, and this one for sure would be something from the Star Wars. But this was maybe 5-6 years ago, and of course now we know that this, okay, I can't go forward. Okay, that this is really a virus referred to as coronavirus, which led to coronavirus disease 2019, affectionately known as COVID-19. And COVID-19, as we all know, is introduced by this virus, SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome Coronavirus 2, as opposed to the first one. And the episode started in Wuhan, China in December 2019 and was declared world pandemic in 2020, as you know. And to date, more than 6.4 million people have succumbed to this disease. Now, this is a very serious disease, but if you look at the overall picture, the world population being 6.4 billion roughly, so it's like 1 in 1,000, which is not that big of a concern, but still for those that succumb and their relatives and all that, even 1 is a concern, of course. So in relation to viruses, these are submicroscopic infectious agents. They are really not living organisms because they do not fall into biological definition of a live entity, but they contain DNA or RNA that are the genetic machinery, and they are really the most opportunistic parasites in a way because they need a host to get into, and if they are RNA viruses, they get into the endoplasmic reticulum, Golgi apparatus, and replicate themselves enormously. And if they are DNA, they hijack the DNA machinery of the cells and replicate themselves. Nonetheless, they either case, once they have done this, then you get the infection. And as far as coronavirus is concerned, we see the name comes from this crown-like spike proteins, and it's a beautiful kind of virus with the RNA here. So again, I would like to emphasize that having the crown and the beauty doesn't necessarily translate into kindness, as we see with the 4 million deaths. Now, viruses have two ways of entering the cell. One is through a receptor, and the other is through endocytotic mechanism that they fuse with the membrane. But either way, they're going to replicate once they are inside. And for coronavirus, they use primarily the receptors, and we'll discuss that in detail. That's the ACE2 receptor. So, as you know, the symptoms appear in about 2 to 14 days, many different flu-like symptoms really originally, except you have here also dry cough and loss of taste or smell that points to coronavirus in a way. But fatigue, muscle ache, headache, sore throat, these are what you see in common flu as well. But you have emergency warnings, like if you have problem breathing and some other central symptoms, confusion, and other signs and symptoms, chest pain, etc. Then you also have some comorbid condition that can enhance this demise. One and most important is old age. And the reason is because with old age, everything kind of, as I can attest to it, goes down the drain in a way. Everything gets weakened, like a very old car, in a way that you have the immune system, muscle, everything. And you also have comorbidity with metabolic diseases such as obesity, hypertension, diabetes, and I have here the topic of our talk, smoking, with double question mark, and that's what we're going to discuss in detail. Nowadays, a major culprit in many neuropsychiatric, neurodegenerative diseases is considered to be hyperinflammation. Neuroinflammation for neurodegenerative diseases, and this is associated usually with oxidative stress. And in terms of COVID-19 specifically, you have what you call cytokine storm, because cytokines, when they are overexpressed, can result in hyperinflammation. Inflammation is actually an important point of healing process, but when it becomes hyper or exaggerated, then we get into a big problem. So how did I get into all these? Two very interesting episodes. One, and this is how science and pharmacology work in mysterious ways, really, pharmacology or science in general. So when this thing was hitting the news and all that, we just saw this paper in early 2020 by Australian researchers that they said ivermectin inhibits the replications of this virus in vitro. Wow, this was if this is inhibiting replication, why not immediately start it in clinical trial or people? And I got even more interested because with my right-hand person, Brooke Getachew, we'll hear more about him later, we had worked with moxidectin, which is a cousin of ivermectin, and we had looked at its interaction with gut microbiota. We had looked at its antidepressant effect, and I called my colleague Darrell Davis at USC, who had given us actually moxidectin to work with, and I asked him about what's the possibility of getting this ivermectin into clinic. He said, I'm getting calls from everyone telling me about the same thing. We don't have the capacity of doing this thing, but so I started looking. I called my friend Joe Hannig at FDA, asked him, what do you think? He said, look, my friend, you might get into liability if you push this too much, because if it's not FDA approved for these specific things and you are going to be using it off-label, there might be some problem. Anyways, make long story short, with a colleague in Chile, we looked a little more into ivermectin, and since 2020, there have been more than 200 papers on this drug. Bottom line is that in some cases with clinical trials and whatnot, it would have some beneficial effects. So the second episode of reason how I got into this was because of what Elisha mentioned in the beginning, my interest in substance use disorders, specifically nicotine and alcohol, but nicotine and smoking. And then there was this knowledge that basically drugs of abuse can impair the immune system. And as I mentioned before, reduced immunity can cause increased susceptibility to COVID-19. So, well, this might be something worth looking into. And more interestingly, it was initially observed that smokers might have some protection against the COVID-19. So this was a paradox, because smoking can reduce illness, but then you have less presentation of this disease. Honestly, this wasn't a big paradox to me, because I had seen a similar case in smoking, and specifically Parkinson's disease, which I'll come to later. So this started us some brainstorming, and this is how science develops with some colleagues, Mickey Ashner at Albert Einstein, Brooke Gutachew, who is in our department, and Robert Copeland, current chair of the department. And we came up with the idea, let's look into this. And eventually we started to explaining this paradox by looking at nicotine properties, and specifically its interaction with the receptor that brings the COVID into the cell, ACE2, and we knew about anti-inflammatory effects of nicotine, that it is a potent anti-inflammatory agent, and it might cause suppression of cytokine storm, and so that would be a good introduction to the topic. So we did the work on this, but before going into the details of that, let me tell you about the receptor for this coronavirus, ACE2. So what is ACE2 that brings the coronavirus into the cell? Whoever has studied physiology knows that the body regulates everything, including the salt and water, and the regulation of salt and water in the body, which is very important for maintaining the pressure and fluid, is by this renin-angiotensin system. So basically, when the blood pressure or fluid volume drop, it signals the kidney to release renin, which acts on angiotensinogen, which comes from the liver, to convert it to angiotensin 1, and ACE, angiotensin converting enzyme, is also might call ACE1, converts angiotensin 1 to angiotensin 2. And this angiotensin 2, by causing release of aldosterone from the outer layer of the adrenal cortex, causes water reabsorption in the kidney, and also by causing vasoconstrictions in the blood vessels, maintains the blood pressure and the fluid volume. So we knew about this system, and in terms of the diagram, you see angiotensin and angiotensinogen, angiotensin 1. Now, angiotensin 2 acts on its receptor, like we said, it causes vasoconstriction. It not only does that, but it has pro-inflammatory effects, pro-thrombotic effects, and so on. However, if you convert angiotensin 2 to a metabolite by this enzyme protein, ACE2, which also acts as a receptor for SARS-CoV-2, you get the opposite effect, because the metabolites of angiotensin 2, by acting at specific receptor, causes the opposite effect as you see. It causes vasodilation, anti-inflammatory, anti-thrombotic. So ACE2 can actually be very helpful in terms of the overall maintenance of these antithrombotic, anti-inflammatory, etc. On one hand, it does act as a receptor, so you would think that too much of ACE, not too much, but a lot of ACE2 would cause more entry of virus. On the other hand, you need this ACE2 to have this anti-inflammatory. So the overall effect of ACE2 is positive. And just to reiterate, or just do a little more physiology on this thing, basically, because angiotensin 2 has all those maleffects, vasoconstriction, hypertension, etc., for hypertensive people, you use ACE inhibitors. And this is a very common drug in hypertension. And lately, in addition to ACE inhibitors, you can have angiotensin receptor blocking drugs, which would have the same thing. And these two are very common first line of intervention with hypertension. So we knew all these, and so we said, how does nicotine interact with ACE2? And based on the interaction of nicotine with ACE2, and the increase in the entry of the virus, some people postulated that nicotine exposure would increase the risk of COVID-19 damage, including neuroinfection, which is a part of the virus getting into the brain. And this is now becoming an issue because of the long-term effect of COVID-19 on central nervous system. But we thought that this doesn't quite jive with the literature and what we know about nicotine in terms of what I mentioned earlier. And I'll talk a little more about that. But to make a long story short, we looked into the interaction of nicotine with ACE2, and what we came up with, and this is part of the paper that we submitted and was accepted in Federation of European Biological System, and has been a controversial paper, but nonetheless been cited, been even on chat, because I'm going to come to this again and again, separating smoking from nicotine. Very, very important. Anyways, we came up with this, that upregulation of ACE2 may actually be beneficial because of its potent vasodilatory, anti-inflammatory, antioxidant properties. And there was one more support for this hypothesis that children, because they have milder COVID symptoms, have higher ACE2. So ACE2 in general can be a protective, and because of the nicotine interaction with this, we would say that nicotine can be beneficial. And in fact, there were a lot more studies on this advocating a clinical trial with nicotine. So again, to reiterate the distinction between smoking and nicotine, smoking is really bad for you, no matter what you might say, even if it has some protective effects initially against COVID-19, or as we'll see later, against Parkinson, but it can cause so much damage that you might not get to reap the benefit of Parkinson, which is an age-related disease, or with COVID-19. In fact, once you get admitted to the hospital, if you have been a smoker, the prognosis is going to be a lot worse because of the compromised respiratory system. So bottom line, nicotine might be good, but smoking is bad. The other potential benefits of ACE2 and nicotine is that both have antithrombotic effects, and we now know that COVID has very direct interaction with coagulation process, and thrombosis is one big element in morbidity of COVID-19. And actually, patients are started on heparin as anticoagulant. And we looked a little more into this with the interaction of COVID, specifically with vitamins D and K. And my colleague, Dr. Bruce Gutachew, will be presenting this in detail in the fall, sometime October, once Elisha establishes that date. So, bottom line, we have this picture. Smoking, definitely bad for you. COVID, definitely bad for you. But nicotine might counteract the effect of both of these and have a beneficial effect in general. I might mention that because of this, not this paper specifically, but so many others, one clinical trial was started with nicotine, and what they used was nicotine patch, and in a specific population. I got to review this paper for Lancet, which unfortunately didn't make it for publication because of some shortfalls. But again, what I want to emphasize, nicotine patch versus other mode of administration of nicotine is a very important topic that I'll come back to again towards the end of the talk. But let me talk a little about nicotine, subject close to my heart, not literally, but scientifically in the past 30 years, basically. We started with nicotine because at the time I had a graduate student has been, was interested in working with me. I was doing something else and was interested in something different from stress and aging, what I was concerned with. And I had seen that mothers who smoke during pregnancy have higher incidence of ADHD kids. So I said, well, this is a potential topic. We can give pregnant rats nicotine, see what happens to the pups, look at their hyperactivity, look at neurobiological basis. And so this got me started in nicotine, and I'm going to give you the synopsis of how we developed this into what we are today. So nicotine is a rather simple structure, can be synthesized easily. It comes from this beautiful flower that leads to the tobacco, and the tobacco nicotine really is why people smoke. It's because of the nicotine in tobacco. So if the best reason for that, if you took the nicotine out of tobacco, people and made this nicotine-less cigarettes, people would quit in 2-3 days. They would not touch it because it doesn't give them the kick of nicotine. Now, nicotine, in addition to being a potent stimulant and highly addictive, has many other very interesting properties, including anxiolytic. People who smoke claim that it calms them down. It has antidepressant effect. In fact, if you look at the population, general population, you would say that there are maybe 20% of people in general smoke. But if you look at a population of depressed patients, you would see 50, 60% of them smokers. And if you looked at the people with psychotic syndromes, even up to 90% might be smokers. And the reason we believe is that both properties of nicotine, antidepressant, antipsychotic, is being used as self-medication. Nicotine might also have cognitive enhancement effect, so this might have some usefulness in Alzheimer's and mild cognitive in cases. Analgesic effect, absolutely. Actually, an analog of nicotine called epibadidine that is derived from the skin of Ecuadorian frogs has the most potent analgesic effect, maybe 100 times more than opiates. And unfortunately, it cannot be clinically used because of its side effect. But anyways, Dr. Martha Davila might know more about this than I do, actually. And it has anti-inflammatory. That's why in inflammatory diseases like ulcerative colitis, it might be helpful. And as I mentioned, with COVID-19, because of the cytokine storm and the inflammation, it might be helpful there. And one thing we have been really involved with is the neuroprotective effects of nicotine that I would like to share with you in the next few slides. But in addition to all these, nicotine might have therapeutic potential. I mentioned already ADHD because of its being a stimulant, Tourette syndrome, obsessive compulsive syndrome, and I mentioned ulcerative colitis as well. So a lot of potential good for nicotine, including Parkinson's disease. Now, I mentioned that the incidence of Parkinson's disease is lower in smokers. And this has been shown by epidemiological studies ever. Very, very important point of caution. Please don't go start smoking to delay Parkinson's, because like as mentioned, you might not get even to that age to reap the benefit of this nicotine. Now, but nicotine might have a therapeutic potential in COVID. And in fact, with Dr. Copeland, we have a patent pending for use of nicotine in Parkinson's disease. Currently, we have some medications for Parkinson. As you know, it deals symptoms of movement disorder primarily, but it has also non-movement, specifically depression, which nicotine can also be helpful there, and other symptoms. Currently, what we are using for Parkinson's are drugs that increase the dopamine, because Parkinson's is associated with loss of dopaminergic neurons in specific brain areas called substantia nigra. Unfortunately, this drug replacement, L-Dopa replacing dopamine, lasts only a few years, and then you get dyskinesia and other symptoms that could be worse than the initial. So we need some neuroprotectants to delay the degeneration. And we think nicotine or nicotinic agonists or nicotinic modulators might be such candidates. And we reviewed this in a recent paper in neurotoxicity research last year, basically. So in terms of the PD and COVID-19, COVID-19 can have detrimental effect on CNS, but there is no association of COVID with PD. But because COVID can have some effects on nervous system, it can cause the susceptibility to COVID-19 even more and accelerate the progression of the disease. And this was fully reviewed just a month ago. So what is nicotine? We discussed, but how does it act? The action of nicotine is mediated through nicotinic receptors. These are ligand-gated ion channels. These receptors, basically, you have some units that combine to make a pore, and once nicotine binds to this receptor, it opens the pore and you get the flow of ions and excitation of the neurons. So this is a very fast acting mechanism for inducing excitation or sometimes inhibition, like for GABA, in the neuron. It's a very fast system as opposed to G protein coupled receptors. G protein coupled receptors are basically 80% of the drugs act on this system. This is very well defined. Basically, the distinction between the two is that the G protein coupled receptor acts through a transmembrane receptor, and once the ligand or drug binds with the receptor, it starts a cascade of events, specifically through cyclic AMP called second messenger, that eventually leads to the excitation, or depending on the second messenger, inhibition of the cell. So this is a relatively slow process in terms of the neuronal activity. It's still very fast, but compared to the ionic channel, it's a slow process. So the system, when we say nicotinic system, the body doesn't make nicotine, of course, to have these receptors. These receptors respond to acetylcholine, which is a very important neurotransmitter, and acetylcholine can act both at ionotropic receptors like nicotinic or muscarinic receptors, which are G protein coupled receptors. Now, the muscarinic receptors also have their own influence, the many different areas, and they have many different receptor subtypes, CNS, heart, smooth muscle, endocrine, etc., etc. But this is not the topic of our discussion, as we are discussing the ionotopic nicotinic receptors, which, thanks to torpedo fish or electric ray, which has abundant number of these nicotinic receptors, this really was the first receptor to be fully characterized because of the abundance and ease to work with. And so we now know a lot about this receptor, all the subunits, distribution in the brain, etc., etc. So basically, these receptors, now we know, are found in neuromuscular junction. All the muscular activity are due to this activity of the acetylcholine at the neuromuscular receptors through the nicotinic receptors. Then you have plenty of these receptors in the autonomic ganglia that control a lot of autonomic activities. And of course, in the central nervous system, in reward pathways, this has to do with the rewarding effects of nicotine, the addictive component, and other parts of the brain that has other functions as well. So basically, at the moment, we know that there are 9 alpha subunits of nicotinic receptors, 3 beta subunits that combine in different forms, and you find different forms of these in neuromuscular junction, in autonomic ganglia, and CNS. So they combine differently. So the subunits that you find in neuromuscular junction is not what you find in ganglia, and it's not the ones that you find in central nervous system. In the central nervous system, the most abundant are alpha 4 beta 2 and alpha 7, which have been studied in detail, but still there is a lot of open area for research to know what receptors do what and try to exploit them therapeutically. As I mentioned, acetylcholine is a very important neurotransmitter, excitatory, has wide distribution throughout the brain. This is a rat brain that shows you various areas, the cell clusters and the trajectories, and same thing in human brain. Basically, one area, what is called septohippocampal system. As you know, hippocampus is very important in information processing, storage, retrieval, and memory area. And so this septohippocampal system and also cholinergic system in the cortex are one of the first neurotransmitters that are damaged in cognitive dysfunction, including Alzheimer's disease. So basically, same like idea with Parkinson's, where we found that there is lack of dopamine, so give L-Dopa to increase dopamine, retrieve the, or alleviate the symptoms. With cholinergic pathways, said, okay, let's give acetylcholine. Well, you can't give acetylcholine, it gets degraded immediately, but acetylcholine precursors like choline, unfortunately, those interventions have not been very successful. What was of some help, very, very modest, was instead of giving some precursor for acetylcholine, because acetylcholine gets degraded very quickly in the synapse by acetylcholinesterase, to give acetylcholinesterase inhibitors as a way of increasing acetylcholine which could then interact with both muscarinic, but really, we think nicotinic receptors to alleviate that cognitive dysfunction. And currently, really what you have for Alzheimer's disease with very, very modest intervention is acetylcholinesterase inhibitor, and the most prominent drug is donepezil, which I am proud to say that my first graduate student, Sharon Richardson, was involved with the development and marketing of this drug long time ago with Eisai Pharmaceuticals. And it was fortunate we got some grants from them to look at the donepezil interaction with other systems, etc. But make long story short, we think that finding some nicotinic agonists here might not only be of help in Parkinson, but also in Alzheimer's. So one very, very important point I want to emphasize is mode of nicotine administration. And this might be critical factors in nicotine's pharmacotherapy. Actually, because of the potential utility of nicotine in Parkinson's, people tried nicotine patch in Parkinson's disease. There were 8, 9 clinical trials, with the exception of one that showed very modest improvement. None of the others worked. And we could have told them this would not work because the mode of administration of nicotine, when you give it by patch, nicotine patch was actually developed for smoking cessation. And it makes sense because when you give the patch, you're getting a continuous steady state release of nicotine that causes desensitization of the receptors and would reduce your craving for nicotine. And then you gradually wean the person from the patch and they would become accustomed to not smoking. Whereas with neurodegenerative diseases, we want the opposite. We want to stimulate the nicotinic receptors in the same way, kind of, that the smokers do. The smokers are not having a constant steady state. They smoke, they take a puff, and they have a break between each puff, and they have breaks in between cigarettes and overnight withdrawal, and then the next day. So what we are suggesting is pulsatile administration of nicotine that has to be considered. And this is what we are having as a pattern with Dr. Copeland, that hopefully we would get it approved. So basically, again, the take-home message is that smoking is hazardous and can be deadly. Nicotine in pure form may be of therapeutic potential in neuropsychiatric. I mentioned depression, psychosis, and neurodegenerative diseases because of its anti-inflammatory effect and other effects, neuroprotective basically, and because of the topic of related to COVID-19 today, inhibition of cytokine storm can be effective, but we still have to wait for clinical trials specifically on this. But I must mention that honestly, the advent of the vaccine and the drugs that were developed, thanks to pharmacology and all the researchers, millions and millions of lives have been saved, in my opinion. So, but as far as nicotinic system is concerned, we can have a lot of opportunities by looking at the specific nicotinic agonists or modulators that would have the beneficial effects of nicotine, which but without the addictive component, which might be a concern. And again, mode of nicotine administration has to be considered, and it is very important. So with that, I want to acknowledge my collaborators at Harvard and outside, Michael Ashner specifically with the presentation that I had today, but a lot of people at Harvard, colleagues in anatomy, Dr. Chuk and Manai physiology, my graduate students and postdocs, and the support by NIGMS, NIAAA, and Harvard University resources. And with that, I want to thank you for your attention. And related to Elisha again.
Yes, thank you very much, Dr. Tizabi. We did have one question that came up in the chat. Joanne Allard asked, in general, is there higher expression of ACE2 in children and or young adults?
I think it has been shown in children, and that's why they say the susceptibility is less. Young adults, I honestly don't know, but I know because there was a concern with the vaccine efficacy in children as well. So that was one of the things that was investigated initially with children.
I see. Thank you very much for your question, Ms. Allard. Does anyone else have any other questions?
Or comments?
Or comments?
I do have a question.
Martha, yes. You missed my mentioning your name, I think. And I refer people who have questions about analgesic effects of nicotine and specifically epipatidine to you.
Thank you. So my question is the following. So I thank you for the talk. It was really interesting. And I actually had read your paper, so that was really nice. I do have a question regarding, how do you marry the fact that you are hypothesizing that nicotine can actually be used therapeutically for certain, for COVID purposes, but on the other hand, earlier studies had shown that primarily in China, smokers were actually the population that had the greatest negative effects, and that the smokers had, for whatever reason, more vulnerability to get the SARS infection, and their COVID symptoms were actually worse than non-smokers.
So there were a lot of controversies on that. And actually, what started the whole ball rolling was some opposite observation, in that the incidence or the presentation of COVID patients, there were less smokers presenting with COVID symptoms. But once a smoker was admitted to the hospital because of the compromised respiratory condition, they would succumb more easily. So the smokers, because of that nicotine, were thought to have some protection. But once you got in the hospital, because of the compromised respiratory, they were more susceptible to succumbing to COVID. So, and honestly, this was a somehow controversial, and up to now, it's a little not quite settled, but I think it's accepted that nicotine, not smokers, remember, again, we're separating these two, that because of the nicotine interaction with ACE2 and other things, might actually be beneficial. So it has to be distinguished between smokers, and the epidemiological studies have had different things, but in general, overall was that the incidence or the presentation with smokers is less.
Okay, but so nicotine actually does act on all the subtypes of nicotinic receptors, and so that may actually be also a combination of effects as opposed to direct effects on the nicotinic receptors that you are talking about, perhaps?
Yes, exactly. I think in terms of the peripheral system and central, absolutely. What I mentioned was a very brief overall presentation saying that in CNS, and of course you know better than me that you have the others, alpha 3, alpha 9, that are involved in the addictive properties and other things. But I mentioned alpha 4 beta 2 and alpha 7 as ones that have been most heavily studied. Yes, you are absolutely right. We have to see what specific receptors are doing the trick.
And also, Dr. Abbas, I'm not sure if anybody is asking for questions, but can I just say one more thing?
Please.
Yes.
Okay.
Yes, sure.
So Dr. Abbas just asked whether it would be possible for nicotine to trigger activation of immune cells. And yes, the immune cells do have nicotinic receptors on them. So that's another possibility that you're not only interacting with nicotinic receptors in the periphery, you're also interacting with nicotinic receptors, perhaps even in the brain, but also in the immune cells. And we have to really sort all those things out before you can actually make nicotine gum or treatment more available. I think we have to sort all that stuff out. Don't you think, Dr. Tizabi?
Yes, Dr. Abbas, that's a very good question. In fact, you are now working with inflammation and cancer and stuff like that, and immune system specifically. And I think there is a lot of room for nicotinic system. Immunology is a terrific subject and full of puzzles. And I think once you find some lead drugs that can affect specific, I mentioned ulcerative colitis, which is, and really, the same thing with the CNS, multiple sclerosis, which is an autoimmune. Even there, nicotine might have some beneficial effect, but it has to be looked at. It's just a hypothesis at this point.
Thank you.
Sure. So
Thank you.
maybe we can talk about collaborative work. I know Dr. Davila is interested in. Let's brainstorm. Brainstorming in science is always the trick.
Absolutely. If I may, just have a comment here, because I've been studying genetic variation in serotonin receptors on immune cells. So we know that also immune cells, have receptors for serotonin, and we found some interesting, data on that. So absolutely, this is a very, very excellent, connection between, let's say, neurotransmitters and whatever comes, to attach to these neurotransmitters and the immune function. So there could be a relationship between, nicotine and activation immune cells, and maybe protection from COVID. So absolutely, this is something, we need to look at too, but this is excellent. Thank you.
Yeah, I might add to that Brooke is an avid investigator, tries to connect the dots. And we saw actually something with nicotinic and serotonergic system. And, of course, serotonin has also maybe 15 receptor subtypes, but one specific one, 5-HT3, I believe, is ligand-gated ion channel. So, and the ligand-gated receptors interact with each other. So it is very likely that we might have a common mechanism there with serotonin nicotine and the immune system. Yeah, definitely. Let's. I see Brooke has raised his hand.
Yeah, the 5-HT3 receptor is not only like ion channel, but it also have sequence homology similar to the nicotinic subtypes. So there's some connection there. So, and it will be worth investigating what they have, if their expression is different in immuno cells, and if there is some manipulation that could be done, not necessarily with nicotine, but you could use nicotinic agents such as varacloline, which is a partial agonist that's used for smoking cessation, or other nicotinic agents that come to play.
Someone wants my screen to be shared. Okay. But I think Elisha has the screen, right, at the moment?
Yes. Oh, you have it.
Now I have it.
Yes. Yes. I believe Dr. Davila Garcia commented that we have shown alpha receptors in T cells, and she dropped the link to the journal article.
Fetal cells, yes. And that might be the link between mothers smoking during pregnancy and having a higher number of ADHD kids, basically, because once you manipulate those receptors prenatally, once the baby is born, then the development is already kind of interfered with and can lead to consequences. The same like fetal alcohol syndrome, et cetera, et cetera. Yeah, thanks, Martha.
Dr. Getachew, did you have another question or comment? I see your hand is raised.
No, I don't actually.
We're gonna put it down.
Are there any other questions for Dr. Tizabi? Any comments?
So I just want to thank everybody again for participating, and Elisha in particular for making this possible, and looking forward to more of these interactions.
Absolutely. Thank you very much, Dr. Tizabi, for taking the time to tell us about your research and answer some questions. Thank you to everyone for joining us and participating in a discussion. You can email us at rcmi-cec@howard.edu if you have any further questions or comments about today's presentation, or if you'd like to learn more about how we can support your research projects. Please join us for our next Journal Club meetings as we address topics such as COVID-19 induced anosmia recovery using phytochemicals and essential oils, as well as the problem of coagulation in COVID-19 and the use of vitamins with Dr. Getachew, who is with us here today. Thank you for joining us. Until next time, take care, everyone, and enjoy the rest of your day.
Thank you. Bye-bye.
Thank you.